Hi students, I welcome you all to the very first biology class conducted by Manorama Horizon. In this class today we'll be discussing the chapter The Fundamental Unit of Life. This is the very first chapter in biology. So let's start with the chapter. What do you understand from this image? Just guess, what do you understand or what do you understand by seeing this image? So here you can see that there's a boy and the boy is visualizing something through an instrument. Okay. Now what is this instrument? What is he using here to visualize things? Yes, he is using a microscope. So what is he using? He is using a microscope. Now why is he using a microscope? Microscopes are instruments that are used for visualizing very small things, okay? The things which are not visible from our naked eyes are used to visualize by microscope. Now, if you see, this is a compound microscope. Compound microscope, or we call it as light microscope, because the source of light is either sun or artificial light, okay? And that light is captured by the mirror. So this mirror helps to capture the light. Now in this microscope, usually the specimens are placed here in the stage. Okay, so that is the stage where the specimen to be visualized are placed. So this is the stage. Okay, so in the stage, the specimen to be visualized are placed. Now here you can see this part is the eyepiece, and from the eyepiece we will be able to visualize the specimen. Now here you will be having lot of lenses. These lenses are known as objective lenses. Okay, so these lenses are known as objective lenses. These objective lenses, they help to magnify the specimen by 10, 40 or even 100 times. Okay. Now this microscope, that is the compound microscope, is the most commonly used microscope in our schools, even in colleges. So this microscope helps us to visualize cells, nucleus. If we well strain the nucleus, we will be able to see the nucleus. But this microscope doesn't help us to visualize still smaller parts of the cell, such as cell organelle or the structure of cell membrane. So for that, we require a microscope called electron microscope. Okay. Now electron microscope, what it does is, it magnifies the image thousands and thousands of times. So we'll be able to see every molecular characteristics or molecular elements which are present in a cell or any other objects. Okay. So is it clear? Now first, today we are going to study about cell. The first cell was visualized by Antoine Leeuwenhoek. He has self-designed his microscope, and using his self-designed microscope, he first visualized the cell. But the term cell was given by Robert Hooke. Okay, Robert Hooke was a person who visualized Koch cells and gave the term cell. Okay, cell means here small compartments. Cell means small compartments. Now, when Robert Hooke saw the Koch cells, he was able to see small compartment like a prison, okay, of a jail, or so a small room he was able to see, and he named it as cell. Later on this name was taken as to specify all the fundamental unit of life, we use this term. Okay, so Robert Hooke was the first person who termed that living unit as cell. Okay, now let's study the different parts of the cell. Before going into the different parts of the cell, we'll see the different types of cell which are present in our body. Now if you see here, these are the different types of cells in our body. Now these different types of cell help us to come into two conclusions. Okay, these different types of cells help us to formulate two conclusions. So what are that two conclusions? Can you name what can be the two conclusions by looking the different types of cells? Yes, the two conclusion here is, one, cells are of different size. Okay, you can see different size, as well as cells are of different shapes. Okay, so the two conclusion that here we derive from the different types of cells are, cells are of different sizes and shape. You can see a neuron cell is entirely different from a egg cell or an ovum. Okay, so neuron cell, you can see it's long, branched, whereas a ovum is a spherical cell. Okay, so by seeing the different types of cell, we can derive that cells are of different shape and size. Now, not only here, you can see it in every animals, in plants, in every organism, except this unicellular organism, you will be finding different types of cells. Okay. Now, we got to know that there are different types of cells in our body. There are different types of cells in plants' body, in animals' body. Now, we'll see what are the parts of cells. Okay. So, let's see the different parts of the cell. So, first let's start with the cell membrane. Now, here you can see here cell membrane. If you see an animal cell, this is an animal cell, okay, and this one is a plant cell. So, if you see an animal cell, you will be able to see the limiting membrane. The limiting membrane of an animal cell is cell membrane, or we call it as plasma membrane. It's another name of cell membrane. But in case of plant cell, if you see a plant cell has an outer wall, okay, an extra wall is present, and inside that you will be finding the membrane, cell membrane. Okay, so for animal cell it is a limiting membrane, but for plant cell it is not a limiting membrane because there is a cell wall present outside the cell membrane. Okay. Now, what is this cell membrane made up of? Just think, what is this cell membrane made up of? We can see here the cell membrane is made up of proteins and lipids. Okay. So, lipids are nothing but fats. Okay. So, cell membrane are made up of proteins and lipid. Irrespective whether it is plant cell or animal cell, the cell membrane is always made up of protein and lipid. So, usually there is a question asked, what is the composition of a cell membrane? So, at that time you need to write it as proteins and lipids is the composition of a cell membrane. Now, this cell membrane has particular function. The major function of a cell membrane is semi-permeability. Okay, it helps in the movement of substances in and out of the cell. Okay, it helps in the movement of substances in and out of the cell, but not all substances. Okay, so here they have, you can see a term is used, that is selectively permeable or semi-permeable. Okay, so the membrane is selectively, instead of saying semi-permeable, we may say it more precisely as selectively permeable. Now, what is meant by selectively permeable? Selectively permeable means it allows only selected substance, whatever is required, only those substances are allowed to pass in and out of the membrane. See, for example, if I am taking a bucket here, just imagine I have taken a bucket here. I have separated this bucket by a membrane. Now, I have here some molecules, green molecules, okay, some blue molecules. Now, this membrane, just imagine this membrane is the cell membrane. So, if the cell membrane require only blue molecule to be passed to the other side, it will allow only blue molecule to pass, not the green molecules. Okay, so it will allow only blue molecules to pass here. So whatever is required by the cell membrane, whatever is not required by the cell membrane, only those substances are sent in and out. Okay. So hence the term selectively permeable. So cell membrane is selectively permeable. So you have to remember it is made up of protein and lipids and it is selectively permeable. One more important point about cell membrane is it is present in every organism. If plants have cell wall, then too they have cell membrane. Animals also have cell membrane. Okay, so it is present in all organisms. Even a bacteria also has cell membrane. So cell membrane is universal to all organisms. Next we'll see the difference between diffusion and osmosis. Very important difference you need to know, because in cell membrane, if some molecules are passing through a cell membrane, we say it as osmosis. So why do we say it as osmosis and not diffusion? Knowing this is very important. In diffusion, you can clearly see that there are molecules. Okay. So these molecules are present in a solvent. Now what happens in diffusion is the molecules, that is the solute, those solute where it is concentrated in one region, it start to move and equally get divided in the overall solvent. That is known as diffusion. Okay, so diffusion means the movement of solute molecules from high concentration to low concentration. So here we saw that the concentration was high and it moved to the low concentration and got equally spread. Just imagine your mom is cooking biryani. Okay, you get a smell even it's sitting in the living room, right? So how do you get the smell? So that's because of diffusion. Even the spray, if you just spray near the corner of your room, you will be finding within seconds the whole room will be giving a sweet smell. Okay, will produce a sweet smell. That's because from the higher concentration, the fragrance or the spray molecules has moved to the lower concentration. Now coming to osmosis, here what happens is, here the solute molecule moves. Okay, sorry. In osmosis, the solvent molecule moves. Okay, not the solute molecule, the solvent molecule moves. Now here you can see in the first image, you can see that there is concentration of solute in one side and there is less number of solute in the other side. Okay, so when the solute is less, the solvent is more. So solute is less, the solvent is high. So what will happen here? Osmosis is the movement of solvent from higher concentration to lower concentration. So here you can see here the solute is low and the solvent is high. So the water molecule will move from high solvent region to low solvent region, and the movement take place through a membrane. We call that membrane as semi-permeable membrane. Now you can see here after osmosis, you can see in both the side the solute and solvent concentration has become equal. Okay, so that is osmosis. So here in diffusion we have seen that the movement of solute molecule is taking place from a region of higher concentration to a region of lower concentration, whereas in osmosis we can see that the movement of solvent is taking place from a region of higher concentration to a region of lower concentration. Okay, so is it clear? Is it clear what is happening between diffusion and osmosis? Now just look at this image. Now here 3 different cells were placed in 3 different types of solution. The first cell was placed in hypertonic solution, the second was placed in isotonic, and the third was placed in hypotonic solution. Now when the cell was placed in hypertonic solution, what is meant by hypertonic solution? A solution that contain more amount of solute and less amount of solvent. So it's a solution, hypertonic solution is a solution where more amount of solute is present and less amount of solvent is present. So at that time what happened? The cell loses all the water. Exosmosis take place. In isotonic solution, both the side, the concentration is same. So, no movement of water take place and the cell remain intact. Okay. Whereas in hypotonic solution, the movement of water take place inside the cell. Endosmosis occurs. Because of that, the cell become turgid. Okay, the cell become turgid. So, in hypertonic solution, the cell become plasmolized, all the water goes out. In isotonic solution, the cell does not become, any change is not noticed in isotonic solution. Whereas in hypertonic solution, the cell become turgid. Okay. Next portion, that is, we'll solve some of the questions here. Okay. Now, the first question, give the difference between diffusion and osmosis. I think now you know the difference between diffusion and osmosis. Okay. One is the movement of solute from higher concentration to lower concentration, whereas other is the movement of solvent from higher concentration to lower concentration. Okay. Next, what happens to the cell if it is placed in hypertonic solution? What happens if it is placed in hypertonic solution where the solvent concentration is low? So, what will happen? The water from the cell will move outside. Okay, because of that, the cell will get plasmolized. Now, next, what is the composition of plasma membrane? What is the composition of plasma membrane? It is made up of protein and lipids. Okay. Now, we'll move on to the next part of the cell, that is cell wall. If you see, cell wall is present only in plant cells. Okay, it's not present in animal cell. It is present in plant cells. Even in bacterial cells also it is present. Okay, it is present in plant cells as well as in bacterial cell. It is not present in animal cell. Now, this cell wall, again the question is asked, what is the composition of cell wall? Cell wall is composed of cellulose. Okay, cellulose is made up of glucose. So, cell wall is composed of cellulose and cellulose is made up of glucose. Cell wall is very important for plants because in our condition you can see any of the environmental stress comes, we just run off to the shelter. Okay, we have a shelter, we can walk, we just move on to a safe shelter. But in case of plants, they cannot do that. So, they want a strong cell wall to protect themselves from all the environmental stresses. Okay, so you can see here, provide structural support to the plant. So, first thing, it make the plant hard. Okay, cell wall make the plant hard. It provides structural support to the plants, as well as what it do, it protect the plant from different kinds of stresses. Okay, different kinds of stresses, the environmental stresses, heat, cold, rain. Okay, even the pathogens like bacteria, fungal attack, all this it protect the plants. Okay, so this is done by cell wall. Even when the cells are placed in this hypotonic or isotonic conditions, so what happens in hypotonic condition, the cell get plasmolized. In hypotonic solution, the cell get turgid, but the plant cell doesn't break because of the strong cell wall. In hypotonic solution, if we just place animal cell, the animal cell may burst actually. Okay, after absorbing lot amount of water, it burst. But the plant cell will not burst because of the presence of cell wall. Okay. Now, next we'll study about nucleus. Nucleus is the control and coordinator of the cell. Okay, we can say that it's the brain of the cell, the most important organ of a cell. In plant cell, if you see, nucleus are not at the center. This is an animal cell where you can see nucleus at the center. But in plant cell, the nucleus is seen at the periphery. Why it is so, we'll learn it later. So, if you just see the structure of nucleus, as I told you, it's the main control system of the cell. It has a complicated structure. So, first let's see. You can see here that nucleus has small pores in it. Okay, the membrane of the nucleus has small pores in it. So, this pores help nucleus to communicate with the cytoplasm. Okay, the inner medium of the nucleus as well as the outer cytoplasm communicate with the help of the nuclear pore. Okay, then we have inside the nuclear pore, we have similar to inside the cell, we have a jelly-like substance. Similarly, inside the nucleus, we have a jelly-like substance called nucleoplasm. Because it is inside the cell, we call it as nucleoplasm. Apart from that, we have in the center, we have a circular structure which is known as nucleolus. We have a double membrane layer that covers the whole nucleus, which we call it as nuclear membrane. Apart from that, you can see this thread-like structure. That thread-like structure is known as chromatin reticulum, and it is made up of DNA. Okay, you can see in this image, animal cell is shown here, and inside that, inside the nucleus, there are chromosomes, and these chromosomes are made up of DNA. So DNA are like threads. You just imagine a thread. DNA are like thread. So when we buy thread, we'll be seeing that the threads are usually coiled, okay, coiled on some cardboard cylinders. So similarly, here in the cell, the DNAs are coiled, okay. So that coiled DNA structure is known as chromosome, okay. It is known as chromosome. DNA are large unit, they cannot be kept as such, hence they are kept in a very coiled way. So that coiled structures are known as chromosomes, okay. So DNA are the one who actually controls everything, every activity, every metabolic activity of a cell. Now you can see here, controls all the activity occurring in a cell. So nucleus controls all the activity occurring in a cell. Apart from that, nucleus does a very important function that is known as cell division. Nucleus helps in cell division. A cell can divide only because of the nucleus. So first the nucleus divide and then the cell divides, okay. So is it clear? I hope there is no doubt related to nucleus, okay. Now let's move to the different cell organelles, okay. Before moving into the cell organelle, we'll just understand what is the difference between a eukaryote and a prokaryote. Now eukaryote, prokaryote, the meaning itself says karyon means nucleus. Karyon means nucleus, okay. So prokaryon, before nucleus. So these organisms are primitive, okay. They do not have a proper nucleus. You can see in this image, here eukaryotic cell has a nucleus, whereas a prokaryotic cell has only DNA scattered in the cytoplasm, okay. There is no nucleus here, there is no membrane, nuclear membrane is not present. This structure we call it as nucleoid, but there is no nucleus, okay. So prokaryote, before the nucleus, primitive nucleus we say it as. And U means true, eukaryote means true nucleus. So that's the major difference between prokaryote and eukaryotes, okay. Apart from that, they have, eukaryotes have cell organelle, they lack cell organelle, okay. Only ribosomes are present in prokaryotes, and eukaryotes have rest of all the other cell organelles. Now let's move to the cell organelles, okay. So before moving to cell organelles, we'll do some questions, okay. So the first question here is, what is cell wall composed of? We have already learned it is composed of cellulose, okay. The next question is, how is prokaryotes and eukaryotes different from each other? How is prokaryote and eukaryote different from each other? Prokaryotes and eukaryotes. Prokaryotes lack a true nucleus, whereas eukaryotes have true nucleus. Now coming to chromatin fiber condense to form chromosomes. They condense to form chromosomes. Give the function of a nucleus. So you can give the function of a nucleus, okay. It controls all the activity of a cell. It helps in cell division. Now we'll be seeing the different cell organelles. Now what are the different cell organelles? Can you tell me what are the different cell organelles? We have seen in the previous picture that a eukaryotic cell contains different types of organelles. So we'll quickly go into each of those organelles. Okay, so first we'll cover endoplasmic reticulum. Now if you see, these are very complex structure. You'll be finding these kind of structures. Okay, so these kind of structures are endoplasmic reticulum. Reticulum means network. Okay, now here endoplasmic reticulum is usually seen attached with the nucleus. It is usually seen attached with the nucleus. Apart from that, you can see the endoplasmic reticulum. Some of the endoplasmic reticulum has ribosomes attached on their surface. So some has ribosome attached on the surface, some do not have ribosomes attached on their surface. So accordingly, endoplasmic reticulums are divided into two. One that has ribosome attached on the surface. So if ribosomes are attached on the surface is not smooth. So we call it as rough endoplasmic reticulum. In short, we call it as RER. And then in some of the endoplasmic reticulum, we'll be finding that ribosomes are not attached. They are known as smooth endoplasmic reticulum. Okay, SER. Now, ribosome, what it does, it helps in protein synthesis. So, the endoplasmic reticulum that are seen attached with the ribosomes helps in protein synthesis. What does it do? It helps in protein synthesis. Whereas the smooth endoplasmic reticulum, because they are not attached to ribosome, they help in the formation of lipids. Okay, they help in the formation of lipids. And do you remember what I said at the start of cell membrane? I said that cell membrane is composed of proteins and lipids. Hence, endoplasmic reticulum helps in the formation of cell membrane. We say that term as biogenesis of membrane. Okay. So now I have already told you that RER helps in protein synthesis. Okay. And smooth endoplasmic reticulum helps in the formation of fats, helps in the formation of lipids. Okay. Even smooth endoplasmic reticulum act as a detoxifying organ, okay, organelle. So it is found in liver. Most smooth endoplasmic reticulums are more active inside liver because all the food we consume, all the toxics that reaches the liver is usually detoxified by smooth endoplasmic reticulum. And then I told you it helps in membrane biogenesis. Now next we'll see the other organelle, that is Golgi complex. So if you see Golgi complex, it got its name by the scientist who has discovered it. So this was discovered by Golgi, okay, Camillo Golgi has discovered this organelle. Now this organelle, whatever endoplasmic reticulum does, the packaging is done by this organelle. Okay, so what this organelle does, this organelle helps in packaging and dispatching of materials, okay, that is the proteins or other lipids, fats and all, whatever is produced by endoplasmic reticulum is packaged and dispatched by Golgi bodies. Golgi bodies also help in the formation of complex sugars. What they do is they combine the simple sugar and form complex sugars. Okay, apart from that, they helps in the formation of lysosomes. Okay, they helps in the formation of lysosomes. So next we'll be seeing what are lysosomes. So lysosomes are small circular organelle. Okay, you'll be finding a circular organelle, and in this organelle you will be finding lots of digestive enzymes. So what these lysosomes do is, whatever damaged materials reaches to it, it helps to digest those damaged materials. Even if the cell has got damaged, okay, the cell wall has broken or the cell membrane got damaged, in that condition, this lysosome bursts and release all its digestive enzyme all over the cell and helps in the digestion of the whole cell. Hence, these lysosomes are known as suicide bags of a cell. Okay, so they are known as the suicide bags of a cell because they help to digest the whole of the cell. Their own cell they help to digest. So they are filled with digestive enzymes. Whatever damaged organelle or any other waste substances, they just digest it. Okay, they are known as the suicide bag of the cell. So we have already covered endoplasmic reticulum, Golgi apparatus and lysosome. We'll now move to the next organelle, that is mitochondria. This organelle might be very famous among you all, right? Now just imagine, to walk, to run, what do we require? For any of the physical activity to be done, even to work or to think, we require energy. And that energy is provided by this organelle. Okay, mitochondria is the organelle in our body that helps to provide energy. Okay, like electricity is used to run a fan or to give the power to many other equipments. Similarly, mitochondria is needed to give power to our body. So they are the powerhouse of the cell. They generate the power, they generate the energy. Now you can see that mitochondria is made up of two membranes. You can see here, mitochondria is made up of two membranes. Usually the first membrane, the outer membrane of the mitochondria is continuous. Okay, whereas the inner membrane, you will be finding lot of folds in the inner membrane of the mitochondria. You can see here in the inner membrane, you can find lot of folds. So this folds helps in the synthesis of ATP. What is ATP? ATP is the energy currency of the cell. It has energy trapped in it. Okay, they are known as adenosine triphosphate. They have energy trapped in it. Okay, so this inner membrane of the mitochondria help in the synthesis of ATP. Now apart from that, they have their own DNA. We have seen that nucleus has their own DNA. Mitochondria also have their own DNA. Okay. Now coming to the next organelle, that is plastid. Another organelle which is seen only in plant. We have seen that one part, cell wall, is seen only in plant. It is seen in bacteria also. But this organelle is seen only in plant. Okay, the plastids are organelle which are seen only in plants. Okay, now there are three kinds of plastids. You can see from the image itself, you can understand that there are three kinds of plastids. One is chloroplast, which is green in color, which has chlorophyll in it, hence it is green in color. Chromoplast, which is colorful plastid. It is of different color, yellow, brown, orange, okay, all different colors. Leucoplast, you will not find any color. It stored food in it, okay, like protein, fat, starch, all these things are stored. If you eat a potato, if you take a potato has more of leucoplast, okay, there is no color, it stored starch in it. If you take a flower, if you take hibiscus flower, the petals are brightly colored, they have more of chromoplast. If you take the leafy vegetables, they are green color, okay, they have more of chloroplast. So the different types of plastids do different kinds of function. Now they also similar to mitochondria have their own DNA in them. Okay. Now next we'll see the last organelle, the last cell organelle, that is vacuole. Now vacuole, as I told you when I was talking about nucleus, I told you that the plant's nucleus are shifted to the periphery, whereas animal nucleus are seen at the center. The plant's nucleus are shifted to the periphery due to the big vacuole present in the plant. Okay, so in plant you will be finding a large vacuole, whereas in animals you will be finding very small vacuole, or sometimes you will not find any vacuole at all. So vacuole, what they do, they store waste materials, cell sap, okay, lot of sugars, proteins, all these things are stored in this vacuole. Okay, so vacuole, they store cell sap. They are seen, they are large in plant cells and small in animal cell. They have only one membrane, that is known as tonoplast. Okay. Now in amoebas, in case of amoeba, you can see they have different types of vacuole. They have food vacuole, okay, where they digest the food. They have contractile vacuole that stored water and waste material, okay. So this kind of different vacuoles are present in amoeba. Now this was about the different organelles. Now let's try to solve some questions. Now if you can see this cell, it has been asked label A and B. So what is A? You can see the invagination, right? By that you can say it's a mitochondria. And what is B? B is a vacuole. What is the function of A and B? Function of A and B: A, powerhouse of the cell, generates power. B, stores all the excess of water or all the excretory substance in them, okay. Now here you can see plastids. We have studied chloroplast, chromoplast, leucoplast. Chromoplast give color. Leucoplast, what it does? Store food. And chloroplast helps in photosynthesis, okay. Now we have some other questions. Which organelle is known as the suicide bag of the cell? Which organelle? It is lysosome. Very good. Then dash helps in membrane biogenesis. Dash helps in membrane biogenesis. Endoplasmic reticulum. What is the function of vacuoles in amoeba? So we have seen that there are two functions. One is food vacuole, which help to digest food, and the other one is contractile vacuole. Then we have, why are vacuoles large in plants and small in animals? We know that in animals they are digestive organs. We know we excrete food. Whatever food we take in, we excrete the waste material, but plants do not have that. So plants store all their waste materials inside vacuoles, hence large vacuoles are present in plants, okay. Next, the last topic is cell division. Now, cell division means dividing of cell, okay, from one cell dividing into two or more cells. That is known as cell division. Usually in animals, in plants, in all organisms, a cell is formed from a pre-existing cell, okay. Now, cell, if you just see, cell division is of two types. One is mitosis and the other one is meiosis. In mitosis, if you see, one cell, that is one parent cell, divides and forms two cells, okay, forms two daughter cells, and you can see the parent cell will be of 2n and the daughter cell will also be of 2n. It's an equational division. It occurs only during growth, development, okay, during this time this division occurs. When we fall, we have a cut, this division take place in our body. Then there is another division, that is meiosis. Here what happened, one parent cell, that is 2n parent cell, produce 4 daughter cells, and these daughter cells are haploid, means it has reduced itself. This division is reductional division. It takes place only in reproductive cell, not in all the cell, it takes place only in reproductive cells, okay, like during gamete formation. So two types of cell division occurs in our body. One is mitosis and the other one is meiosis. Now mitosis division takes place during growth, okay, whereas meiosis division takes place during reproduction, okay. So this was about the chapter. I hope any doubt or anything you can put on your chat box, okay. We'll be trying to solve all the doubts. If you're looking for more such contents, more videos, detailed videos of the class, question bank, previous year question papers, you can subscribe to Manorama Horizon. Let's join to the next class next day.
Manorama Horizon. Learning made simple.